Hey, this is Dr. Ben Weitz, host of the Rational Wellness Podcast. I talk to the leading health and nutrition experts and researchers in the field to bring you the latest in cutting-edge health information. Subscribe to the Rational Wellness Podcast for weekly updates. And to learn more, check out my website, drweitz.com. Thanks for joining me, and let's jump into the podcast. Our topic for tonight is we're taking a deep dive into the GI map stool test. Tonight we have Fabian, PhD of Diagnostic Solutions. And so I'm sorry for everybody who's missing the debate. And I'm sure there's going to be a lot of poop discussed, but we're going to have a much higher level discussion of poop tonight. So Tom, thank you for joining us tonight. And I'm sure we all have a lot of questions about our favorite functional medicine stool test. And so we hope to develop some clinical skills to be able to help better help our patients.
Absolutely. Well, thanks so much, Dr. Ben, for inviting me to join your discussion group. And I look forward to interacting with everyone here and hopefully giving everyone a better understanding of GI map and how to better apply it in clinical practice. All right, so I'm going to go ahead and fire up the PowerPoint here. So first, I'll just share my screen. So tonight, we're going to be focusing on a deep dive into GI map. So this presentation is meant to be flexible since this is a very interactive group. So Dr. Ben, if you have any questions in the meantime, definitely feel free to interrupt, or if anyone has anything that I've gone over they don't really understand, I'm happy to stop and we can just cover whatever we can cover in the amount of time.
Maybe I'll ask just one quick question. Since there's a lot of different types of stool tests that are out there, can you talk about the difference between the technology used for the GI map stool test, which is Quantitative PCR, and how this is different than, say, whole genome sequencing stool testing?
Sure. Yeah, so they are different technologies for different purposes, and you get different types of information. So starting with metagenome sequencing, that's a DNA-based method, as is qPCR, at least the type that GI map is using. So we're looking at detecting the DNA for specific organisms or for genes, specific genes. So for example, in the instance of toxin genes. So you can basically detect any aspect of DNA either way with either type of test. But that's kind of one of the areas where the similarity stops. So metagenomic is used, especially if you look at research, for kind of a broad overview, a survey of the microbiome and the genes that the microbiome encodes to get a sense of the overall community. So there's a lot of ongoing research in that area to figure out, of the hundreds of species that you can potentially detect with that method, what does that actually mean? So that's a lot of ongoing research, and there's really not a whole lot of conclusions we can make definitively about most of the species. A lot of them just have not been well studied yet, although of course we're learning more every day. So that's one of the advantages of sequencing, is you can potentially get kind of a more high level, bigger picture of the ecosystem. But the challenge is, if you want to measure specific clinically relevant organisms, particularly quantitatively, so you can see what their levels are, whether they're increasing or decreasing over time and with treatment, you really need a more quantitative method. And especially when you look at sort of the composition overall of the microbiome, so you have very abundant species, all the way down to species that are barely detectable. So there's a really wide range of abundance levels for different species. Some of the species, in fact, quite a few of them on the low end of that range, are clinically significant. Classic example would be H. pylori. So H. pylori is mostly in the stomach. By the time it's in a stool sample, it's usually pretty low concentration if it's present. So that's difficult to pick up these low abundance species in typical kind of commercial-based sequencing, because the ability to detect these low abundance organisms and to detect them in a way that's at least reasonably quantitative depends a lot on the number of sequencing reads. So you may have heard the term sequencing depth, sequencing reads, and that's expensive to do that. So most of the commercial type testing doesn't really go that deep. So you get a picture of the more abundant ones at the top, and that may include some of the ones in the middle, but oftentimes cannot identify the ones that are on the low abundance end of things very accurately. So that's really where qPCR shines. In fact, it's kind of like a technology that you want to use to find those needles in the haystack. And I'm actually going to go through a few examples here in this presentation. And I do have a couple slides about qPCR as well, where I can elaborate on that just a little bit. But that's one of the main differences. Also, a lot of the metagenome sequencing companies, in fact, I'm not aware of any right now, that also include physiological markers. So to really get some insights clinically into what's going on in the gut, you need to know both sides of the equation, the microbiome, as well as what's going on with digestion, inflammation, and things like that. So you get a more complete picture, even though you have fewer markers, because these types of tests focus on organisms that have been established to be clinically relevant, and also these physiological markers. And then with that careful quantitation, then you can start to really see patterns and trends and things like that. I kind of liken it to, say you're just looking at blood glucose levels on a standard lab test or a hemoglobin test. If all you got was plus or minus, high or low, or sort of high, medium, low, that gives you a kind of a rough idea of what the levels are. But a hemoglobin A1C of 5.6 is very different clinically from, say, 7.5. Although, in some measures, both of those might be considered kind of on the high end. So quantitation really is important, and that's really a big part of what makes a marker more clinically relevant. So that's kind of sort of at a high level, tip of the iceberg, what some of the key differences are in technologies.
Good.
Okay, so this, as you can tell from the title here, so we're going to really focus on clinical insights, how to get some clinical insights out of GI map, particularly by connecting the dots. So that's really where the more advanced users get the most clinically relevant information out of GI map, is learning how to connect those dots on the test with what's going on clinically as well. And of course, the whole point of all that is so you can get better clinical outcomes by really understanding what's going on in the gut. So really, in terms of connecting the dots, you have to know something about the dots and which ones are most important, and then how to connect them. And that really has to do with the microbial ecosystem as well as GI physiology. And those two interact heavily. I'm going to go through a couple examples in this presentation as well, for basically illustrating how those dots are connected and can really yield some great clinical insights. So in terms of qPCR, which is also known as Quantitative PCR or real-time PCR, so sometimes you'll see it as RT-PCR. The purpose of that, and the reason it was developed in research and is also used a lot clinically now, is it's really good for highly accurate identification and quantitation of microbes. I have heard some sort of, when you look at social media and you look at some of the comments that are out there, there's a lot of misunderstanding of methods. So I've heard some sort of competing companies and other prominent folks that are out there in the community say that GI map uses 16S sequencing, which is pretty far from what we actually use. So we actually, again, use qPCR, quantitative PCR, but that its ability to identify something accurately comes down to the choice and the design of the primers. You can design primers that are highly specific for an individual strain, for example. So you can detect right down to the strain level. And for example, that's how we detect pathogenic strains of H. pylori, C. diff, and some of the other pathogens, is based on detecting them at the strain level. So again, it's especially good for low abundance microbes. There's also the issue of absolute versus relative quantitation. It's a little bit technical. I don't want to get into that right now unless anyone has questions, but absolute, you're looking at how much of that organism is present per, say, gram of stool, versus sequencing, which is all relative to what else is there. That can be a problem because if something goes up, that can make it look like something else is going down, when in fact it may be the same level, it's just that it's all relative. So it treats every sample as if it's the same amount. I do have one quick figure here to kind of illustrate that. And that's actually in this figure from this particular article, which is Quantitative Microbiome Profiling Links Gut Community Variation to Microbial Load. So it was published in a prominent journal, Nature, a couple of years ago, where they compared relative versus quantitative approaches and actually found that the absolute quantitation, especially when they looked at Crohn's disease patient, was extremely important, because without looking at absolute quantitation, you miss the whole picture of general decrease in microbes in the gut. And that's a key feature of, for example, Crohn's disease. So I'll show that data here in the next slide. So this is a fairly complicated diagram, but the main point here is on the left is different samples that basically where the data is expressed in a relative sense. So every sample is treated as if it's the same amount, starting amount, and all the different taxa indicated by different colors. So all the different types of microbes make up a certain percentage of that pie. But you don't really know if in person A that the overall levels have gone down, say after antibiotics or after an infection, unless you look at it from an absolute standpoint. And that's what you see here. This is the breakdown, also expressed in terms of total amount per gram. You can see here on the side it says per 10 to the 11 cells per gram. So essentially you can see that there are some individuals that have quite a bit less overall sort of total microbiome in their gut. And they found that to be very clinically significant. So that's one of the big differences between sequencing and qPCR. qPCR is basically looking at things from an absolute standpoint. So big question there is really, why is that important? And again, I kind of alluded to some of those important clinical relevance type aspects, but it really allows you to differentiate low, normal, and high levels at a much more detailed level, so you can identify trends and patterns. And that's a big part of connecting the dots with GI-MAP. And that's also critical for assessing the clinical relevance in your treatment approach, because you want to see if you do a retest, the organism may still be there, but it may be responding well. You may have gone down a couple orders of magnitude in quantity, showing that your approach is working. So that's really where quantitation is important. The other thing that we bring to the table in terms of kind of a differentiator for GI-MAP is how we present our interpretive and educational information. So our staff has a really wide range of expertise, industry experience in functional medicine, clinical testing, several PhDs, et cetera. And so we bring all that kind of cross-disciplinary expertise to the table, particularly with clinical experience mixed with research. So we really focus a lot on the latest research so that we can focus on these interactions. And GI-MAP is really all about this second part of the equation, helping clinicians understand potential interactions between the microbiome and gut physiology. So a key thing to understand here is there's a lot of different aspects of physiology in the gut that really come into play. And once we understand them at a certain level, that can help us understand how to interpret a stool test and what it may mean as far as these underlying issues. So we may, for example, see that, I know you'll see here on the list, pH is one of the major factors throughout the gut that affects the microbiome, and vice versa. Same with oxygen, of course, nutrients from the diet as well, flow rate, digestion, especially stomach acid and enzymes, immune factors, et cetera. So a lot of factors can affect the interaction between the microbiome and our health. And that's really reflected in GI-MAP through 3 common dysbiosis patterns that we've identified over time, just in our overall experience with GI-MAP. So those really boil down to insufficiency dysbiosis, which is a lack of beneficial bacteria. So it's important to understand how to identify that on GI-MAP. Inflammatory dysbiosis, again, very important for a wide variety of diseases, autoimmune and inflammatory diseases. And it's also important to understand how to identify that. And then lastly, digestive dysfunction dysbiosis, which is kind of a generic term that we use because various aspects of digestion may be compromised or reduced and have an effect on the microbiome. And in the microbiome, in some cases, can actually have an effect on digestion. So once again, I'll go through examples of this. And in many patients, they can have a combination of these types of dysbiosis that reflect that interaction between physiological imbalances and the microbiome. So just a note, we do have a resource on our website, downloadable PDF, that lists what markers constitute these different patterns. We will be updating that sometime shortly, within the next couple of weeks or so, and then we'll make that available on our website. But we do have that as a current resource that was created a couple of years ago, so it's just a little bit out of date at this point. The other thing that we really kind of weave into this based on research and how that applies clinically, beyond sort of this holistic, high-level integrative perspective of looking at the whole gut, a big part of that is recognizing upper versus lower GI microbes. So that's something that you may have heard. Some clinicians claim that with stool testing, you can't get any insights at all, except into what's going on in the colon, that you basically don't really understand from a stool sample what's going on in the small intestine, for example. It turns out there's a lot that you can gain from that. You don't get necessarily a completely accurate picture of the composition in compartments that are sort of higher up in the GI tract, but sort of the classic example is H. pylori. We do know that that is detectable in stool, and that certainly yields important clinical information with regards to what may be going on in the stomach. So think of this as kind of a domino effect, that you want to get a bit of an understanding of what's going on higher up in the GI tract, even as high up as the oral cavity. And one of the examples I'll go through that features one of the markers on GI map, for a lot of patients that may actually come from dysbiosis originally in the mouth. And then you want to understand how these physiological imbalances result in its presence in the gut and what it may be doing there. So to really get into this a little bit more, it's important to understand a little bit about the composition of the upper GI microbiome. It's not as well studied as the lower GI, the colon microbiome, but we're learning much more about it, especially in the last few years. Mostly what's dominant in the upper GI from the mouth down to the small intestine are a large class in the Firmicutes phylum called the bacilli. So that's species and groups like Streptococcus, Lactobacillus, Enterococcus, Bacillus, and Staph. We do have these groups on GI map. When you see them overgrown, it's likely that they may reflect imbalances in the upper GI tract. And there's some just basic information about the physiological conditions that they thrive in. They like higher oxygen environments, so they don't like the anaerobic environment in the colon unless the colon environment's been disrupted. They like simple nutrients that are highly available in the small intestine. Bile is in the small intestine, so they're bile tolerant, etc. Also, organisms from the proteobacteria phylum, which we tend to think of more the inflammatory microbes, the dysbiotic microbes, they're much more prevalent as a percentage in the small intestine. So typically they constitute, say, 20% of the microbiome in the small intestine and only 2 to 5% in the large intestine. So proportionally, they're much smaller in the large intestine. A lot of new research has come out on Pseudomonas, which we do have on our test. Actually, I'm going to go back to that. Linking Pseudomonas to inflammation in the duodenum, for example, and linking that to issues with gluten and kind of the broader picture of food sensitivities. They also thrive in a higher oxygen environment, also thrive on simple nutrients like sugars and amino acids. Highly bile tolerant, you can actually find many of these in the bile ducts and in the gallbladder. So they're very bile tolerant. So really, really important group in the upper small intestine. Composition of lower GI microbiome at a high level, the most abundant groups, it's in some ways fairly simple. The Clostridia class really dominates. And that's in the colon, the largest class or largest group in the Firmicutes phylum. And one of the key species is Faecalibacterium prausnitzii, which we do have on GI map as well. That's a major butyrate producer. Again, it's in the Clostridia class. which is the dominant group in the Firmicutes phylum. Then there's a Bacteroidetes group, which is the major group typically in most people in the Bacteroidetes phylum. Akkermansia is a well-known species in the Microbia phylum, which is also one of the key groups in the colon. They thrive in an anaerobic environment. Generally, that whole ecosystem, at least in a healthy individual, thrives on complex fibers, as well as the mucus ecosystem. So the mucus layer in the colon is much thicker than in the small intestine. So that's one of the reasons why there's a more significant mucus ecosystem in the colon. So this is data from Dr. Mark Pimentel's lab that was published just earlier this year. This is just basically showing, you can see at the bottom here on the x-axis, duodenum, jejunum. FD is their term for the furthest distance that the scope could go in the ileum. So it essentially means ileum for the most part, compared to a stool sample. So the take-home point here is the different colors represent the different major groups in the different compartments here, but it's fairly consistent across the small intestine. And then suddenly you have this huge change in the large intestine, which is reflected by the fecal sample. So that's really reflecting that big change in physiological parameters from the small intestine, which again has a higher oxygen content, et cetera, usually lower acid levels, higher pH than the colon. Very, very different environments, particularly because of the fact that it's anaerobic. So that's why anaerobic species thrive there that ferment carbohydrates. So big difference. And that's really key to understanding this sort of localization aspects to interpreting stool tests. And this is the Tyler study mapping the segmental microbiomes in the human small bowel in comparison with stool, the reimagined study. And again, this is a study that was just published earlier this year by Mark Pimentel's lab.
Yeah, we were lucky enough for Dr. Pimentel to join us several months ago.
Oh, you did? Okay, great. Yeah. I'll have to go back and make sure I check that out.
Yeah.
So this is a summary of markers on GMAP based on research that indicates where they're predominant in the GI tract. We just sort of broke it down between upper GI and lower GI, with the upper GI being on the left, and then the lower GI being on the right. So not all species or groups are known in terms of research that's available in terms of where their predominance is. And then in other cases, they may be prevalent in both. But both of these groups here, research supports that they're predominant primarily either in the upper or the lower GI tract. So once again, there's quite a few markers on GMAP that are likely reflecting what's going on in the upper GI. Again, the classic one being H. pylori. Giardia is another well-known microbe that basically thrives in the upper small intestine. The two that I'm going to focus on here just briefly in this presentation are Klebsiella and C. diff. So one example of an organism that's present more commonly in the upper GI and also the oral cavity, and then less prevalent in the lower GI, except in cases of potential inflammation. So as far as stool composition, just to kind of summarize, it primarily reflects the colon composition. So that's sort of intuitive, but also that's what research shows as well. And of course, within the colon, there's a mix of luminal and mucus-associated microbes, with the composition primarily dominated in fecal samples with the luminal microbes. But definitely you get a sense of the mucus-associated microbes such as Akkermansia. It's also dominated by anaerobic fermentative bacteria, in particular the Clostridia class in the Firmicutes phylum, and then the Bacteroidetes genus in the Bacteroidetes phylum. Those are far and away the two most dominant groups in the microbiome in most individuals, at least in Western countries. Microbes from the upper GI tract can be present in stool, but usually in really low numbers. So that's, of course, where qPCR comes in, because we can still get some good insights based on the presence of some of those low abundance type organisms. So once again, this is just sort of back to the figure from the Pimentel study showing the breakdown of these major groups across the small intestine and the stool. Of course, the question is why. We'll get into some of that a bit more as well. Looks like I somehow made a mark there. So really, I think it's important to consider, since we can detect potentially some of these upper GI microbes, some of them are better established as being pretty exclusive to the upper GI tract, again, like H. pylori and Giardia. Pseudomonas, most of the research does indicate that it's likely thriving in the stomach and the upper small intestine. And so far, most studies indicate that it's not present and or not active, usually not active, in the large intestine.
Are you going to have time to go into H. pylori? And if not, can I ask a couple of questions now about H. pylori?
Sure. Yeah. I'm happy to answer questions right now about H. pylori.
Okay. So if we see H. pylori present, but there aren't any virulence factors, essentially, it's my understanding that that's not super important, correct?
So as far as H. pylori's presence without virulence factors, it is important. Apparently, we certainly see a potential clinical impact quite often when it's present, and especially if it's high.
Okay, when we look at the numbers, like the normal says 1.0 e to the third. Now, what's considered high?
High is generally, that's what that cutoff level is indicating. So above that number is considered high. Below that number is considered detected.
Like if it's 5.0 e to the third, is that a little high or a lot high? Is it a lot high if you have like e to the fourth and e to the fifth? That makes it a lot higher, right?
Right. Yeah. I mean, because those are orders of magnitude, but definitely E clinically in the E3 range is high. And it's, of course, you have to take everything into the context of the patient and how that patient's presenting, symptoms that may be related to that. But also we can see a pattern on GI map. And so there's a lot of research showing, for example, that H. pylori, regardless of virulence factors, likely reduces stomach acid in the majority of people that have a chronic infection. And so we have an overall digestive dysfunction pattern that I mentioned briefly earlier that is often associated with H. pylori. And it's one of the ways that we can gauge based on just the test markers and connecting the dots. Does it look like this is having an impact in the ecosystem? And then, of course, the clinician has to decide, is that relevant to the patient's symptoms and how the patient is presenting?
So you're saying if we have a patient with elevated H. pylori, and it seems like it might correspond with the symptoms, it might be worth treating even if there's no virulence factors.
It would be worth considering. A lot of clinicians do, but I think that when it comes to functional integrative type clinicians, a lot of clinicians will often focus more on antimicrobial approaches versus going straight to antibiotics. So it's not a scenario that would be kind of in conventional medicine. They're more looking at the potential virulence in relation to things like potential for stomach cancer, ulcers, and things like that. Some of the more serious outcomes that are tied, the risk for those types of outcomes are tied to the virulence factors. But the more mild sort of to moderate effects based on just depression of stomach acid still can be clinically relevant for patients, but may not warrant sort of complete eradication, for example.
And then are some of the virulence factors more important than others?
Based on research so far, the answer is yes. The two that stand out the most, partly because they've been well researched, but seem to be especially required for the more significant inflammatory outcomes, and that would be CAG-A and VAC-A, VAC-A. Now, it's important to point out, though, that virulence factors tend to work together. And so generally, the more you see on the test, so the more that are positive, the more likely then that could be a more significant scenario.
One more quick question, just in general, in terms of any bacteria or pathogen, or potential overgrowth, when we see the number there, but it's less than the normal, that could still be significant?
Right. Now, again, that's really where clinical judgment comes into play. So,
right, because if it's really low, it has it says DL, right? Like super low.
Exactly. Yeah, that either means it's just not there at all, or it's present at a low level.
Okay.
We often do see that based on interacting with clinicians, that their judgment is, and also based on what we see on the pattern on the test, that E2 can be clinically significant for some patients. Again, mostly in that context of potential for contributing to dysbiosis and reduced digestion.
Okay. Okay, great. Go ahead.
So that's really kind of, it was actually a great time to talk about H. pylori, because that is kind of the classic example when you hear certain folks out there saying you can't really get any information about the upper GI from a stool test. That's simply not true, because H. pylori is sort of the case in point. We can certainly get some great insights into what may be going on in the stomach in that case.
And of course, zonulin gives us some information about the upper bowel as well, right?
Right, exactly. So qPCR, again, just kind of that analogy that it's great at finding these needles in the haystack. And H. pylori is a good example. There are a lot of the metagenomic type tests, metagenomic sequencing, that generally aren't as good at detecting H. pylori, let alone quantitating it. So when you really want to know about a clinically relevant organism, qPCR is a very effective way to assess that. So let's look a little bit at the insufficiency type of dysbiosis. So this is something that probably a lot of clinicians who have worked a lot with GMAP have seen. You may see a really broad insufficiency like we see here for this patient, where there are a lot of markers in the normal bacteria section that are deficient. At the phylum level, that's certainly one of the most important levels to look at, because those two groups together make up the bulk of the microbiome, particularly in the colon. So when they're both deficient, that's telling you the microbiome overall is probably deficient, and then all those beneficial functions that they serve are probably also deficient. It's also great to focus particularly on Akkermansia and Faecalibacterium. Those are two keystone species. So once again, when one or both of those are low, that can be pretty significant in terms of indicating that the colon ecosystem is not so healthy. The other thing I want to note about kind of connecting the dots on GMAP is we do know from lots of research.
Let me just see if I could throw a question in. Dr. Rabar, who's joining us tonight, is an integrative gastroenterologist. Why don't I mute let Dr. Rabar ask a question? Let's see. I'm going to unmute you, Sam. Why don't you go ahead and ask your question? Did I unmute you?
Can you hear me?
Oh, yeah. Now we can hear you.
Okay.
Yes, I can hear you.
Yeah, I mean, actually, I have several questions because we use this test quite frequently. And I kind of use my common sense approach to it. For example, I see sometimes practitioners wanting to treat Klebsiella and Citrobacter pyondi in the stool. And I always question, are these specific pathogens or are these markers of dysbiosis? You know, which means that, you need to look at it as a dysbiotic flora. Or, I mean, like if I got Shigella or something in the stool, I say, okay, I have some specific bug, I got to treat it, or Campylobacter. And, at least from the literature that I see, I don't recognize these as the specific pathogens of the gut. They're indications of this here. So, usually I respond, that treat for maybe SIBO or some other dysbiotic scenarios, but I wouldn't target a specific one. But Dr. Fabian, I wanted to get your opinion on this one without just saying something there.
Absolutely. Yeah. I mean,
and these are two markers that are listed in the section potential autoimmune triggers.
Right. And that actually is one of the examples I'll be covering here in just a little bit. But so that's really why it's considered an opportunistic pathogen. So opportunistic pathogens can exist in the gut without any effects at all, or they can potentially be part of a pathogenic process. So they're not generally recognized, at least in the gut, as being outright pathogens. That picture is probably going to change a little bit with some of the organisms like Klebsiella, where we do see that it's more frequently now associated with things like inflammatory bowel disease, et cetera. So it's, again, in this gray area where it's not thought of as sort of an outright infection, a specific infection like Salmonella, but it is part of a pathogenic process in some cases. So it's kind of in that gray area, and that's a perfect example of where clinical judgment really has to come into play in combination with really understanding how to connect the dots with a test like GI map. So you can really get a sense for, does it look like this is a problem, or does it look like it's, for this patient, probably not much of a problem? And that's well-documented research. Some people can have relatively high levels of Klebsiella and no evidence of inflammation. Other people can have high levels and have inflammation. But the big difference often is the ecosystem. If they don't have as many of the beneficial bacteria, they may be more likely to have inflammation.
Right, and if I may say something, obviously we need to evolve into this to understand if we correct that, whether it reverses the immunological abnormality or not. We can say that this is part of the, autoimmune trigger, but if we fix it, does it fix the autoimmune problem? And that answer obviously is not clear. And so far, I don't think if you just change that bug, you're going to see it, but we have to see how that answer is going to evolve.
Right.
I wanted to ask you a question regarding the H. pylori.
Actually, let me just ask that same question again. Tom, is there any data showing that if we have a stool test that shows potential autoimmune triggers and we reduce that particular bacteria, that we may have a positive effect on their autoimmune disease?
That's a good question. I'd have to go back and kind of dig through some of the research that I have to see what specific studies there are. Mostly to date, what I'm aware of is those are associations. In a few cases, there may be some kind of mechanistic insight suggesting there might be a causal link. But mostly to date, a lot of those are based on associations. I'd have to go back and kind of, again, dig through the research to see if, I do have some studies that specifically talk about whether or not, if they're addressed, does the disease reverse or do some of the symptoms improve? Not entirely sure.
Yeah, I just had a patient today who has ankylosing spondylitis, and he has high levels of Prevotella, and there's data showing that can be directly related to the how the ankylosing spondylitis is created in the body.
Right. Yeah, definitely for some of them, at least it's suspected, and there's some evidence that there might be some kind of molecular mimicry, for example, where the immune system reacts to a protein on the pathogen or the opportunist that then looks very similar to a protein in the body, and then the body develops an immune reaction to those proteins as well. So that's kind of an ongoing thing. I think it's mostly well established for one of the oral microbes, which is Porphyromonas gingivalis. But there's probably a growing list of examples there.
Dr. Fabian, may I ask you a question regarding the H. pylori? We occasionally find this in duodenal aspirate, and it's interesting that even in the stool test, sometimes the marker comes up as red and sometimes it's black. I think, would you agree that the best value is below DL, below the detectable level? So any amount showing would be abnormal regarding whether it's in the black or red zone?
That's a good question. So certainly below detection limit, if you're aiming for eradication or greatly reducing its levels from a therapeutic goal, then certainly that's what you'd be looking at. Now, there's with any sort of microbe, I mean, except for outright pathogens, and H. pylori is kind of more in the opportunistic pathogen category versus something like enterohemorrhagic E. coli, which is an outright pathogen. Those, you generally don't want to see those in the gut at all, of course. Whereas H. pylori, there's some debate as to whether or not complete elimination is always desirable. It really depends on kind of looking at all the risk factors, but some data indicates that there may be some increased risk for other conditions by completely eliminating H. pylori. So what we know mostly from research is that most of these types of opportunists in the gut, like Klebsiella, H. pylori, as long as they're kept in check by a healthy gut and a healthy microbiome, they may not be a problem. So that's kind of where that gray area of low levels for some people may be fine, because maybe they have an overall healthy stomach. They have a lot of lactobacillus, for example, in the stomach, which is definitely one of the known healthy groups in the stomach in a healthy ecosystem. Then they may be fine, and there may be no reason to target H. pylori. But if people are having symptoms that could be ascribed to it, plus we see a signature on the test, there may be other considerations as well. Some clinicians may decide that they want to at least try to reduce that and see if that helps symptoms for that patient.
Does it make sense to also do a H. pylori breath test or antibody test to confirm it?
Some clinicians feel that because it really depends again on the scenario and the risk. Say if you have lots of virulence factors and there's a family history of stomach cancer or anything like that, there may be scenarios where you really do want to make absolutely sure that it's eradicated if you're trying to eradicate it, or even just verifying the levels over time. So there are some clinicians that prefer to do multiple tests just to really get a better handle on it. PCR is very sensitive. So it is often a scenario where we'll see a low level detected where a breath test or a stool antigen test may be negative.
By the way, is PCR ever too sensitive? Is it ever the case that it's picking up things that maybe are no longer there?
That's a highly unlikely scenario as far as past infections. So it's a very different scenario than, say, antibodies, which can linger for a long time. DNA from organisms, especially in the gut, is highly unlikely to linger, partly just from the simple fact that material is moving through within a certain period of time. So if the microbe isn't attached, and to be attached, it has to be alive because it has to actively produce those proteins that allow it to attach. So it's not likely that it would be detectable past a certain relatively short period of time, on the order of days.
Okay. And Dr. Greenberg asked, if the Pseudomonas is high, does that imply that it's overgrown in the small intestine or that it has also colonized the large intestine?
So far, there is no research supporting that it can colonize, or at least colonizes on any sort of frequent basis in the colon. All the research to date supports that it's mostly in the upper GI and the stomach.
Okay.
That seems to be its niche where it thrives. I haven't yet come across a single study. It can be detected, of course. That's how we pick it up. It really doesn't seem to thrive in the colon. In fact, specific studies have looked at that based on transcription, gene expression. And Pseudomonas is one of those that, when you look at gene expression methods, is not detected in the gut, meaning it's not active.
Okay. Yeah, go ahead.
Okay, so kind of coming back to this section, we're talking a little bit about the beneficial bacteria and what to look for when there's a lack of beneficial bacteria. In addition, of course, to the normal bacteria section, you really want to make sure you pay attention to secretory IgA. So we do know that the main stimulus for normal physiological ranges of secretory IgA is the normal commensal microbiome through secretion of butyrate and other products that stimulates the production of secretory IgA. So we see a really strong correlation there. Typically, when there's a lack of at least one of the key groups of beneficial bacteria, we often see low secretory IgA. Clinically, it appears that the lower the number, like we see here, the more likely that is to reflect lack of butyrate producers. So you'll often see, for example, really low secretory IgA below 100 when Faecalibacterium, for example, is also very low. So it's kind of a great overall marker for lack of beneficial bacteria.
So if we see this on a test with a patient who maybe has some other infection or parasite at the same time, would we best be trying to improve their microbiome with probiotics or prebiotics, or would it be better to directly try to stimulate their immune system with colostrum or products like that have an immunostimulatory effect?
A lot of clinicians will focus on both. Building up the microbiome can take time, depending on what's going on. So if you need a more immediate effect, then those types of supplements, Saccharomyces boulardii, L-glutamine, even vitamin A has been shown to be necessary for normal secretory IgA levels. So supplement approaches certainly can help bridge that gap until the beneficial bacteria are recovering. So again, that's a really important marker to pay attention to that has a very strong correlation with lack of beneficial bacteria.
I just wanted to ask about the fecal calprotectin, which is a marker of inflammation. I've had a number of tests where that was normal and not particularly high, but the patient has a lot of gut symptoms and it just seems like their gut is really inflamed. So why would that number be low if their gut is really appears to be inflamed?
That's a good question. So it's a good marker for inflammation, particularly in the lower GI. So if you look at studies on calprotectin, for example, in relation to H. pylori, so calprotectin in stool, there's not much of a correlation. So we know that H. pylori can cause inflammation in the stomach, but that doesn't reflect so much in ability to detect it in stool. So one possibility is that you may have upper GI inflammation if they're inflamed, and that's likely in a lot of cases. So for example, Pseudomonas, lots of new research shows that when that's overgrown, that's likely causing inflammation. Doesn't always mean it is. So again, you want to take into account the big picture. But if it's significantly overgrown, that's a significant possibility that it could be causing inflammation in the upper GI. Candida is known to be inflammatory, often colonizes pretty much anywhere in the upper GI tract, even the oral cavity. So that's one of the major reasons. If they seem to be inflamed and calprotectin looks fine, maybe more in the upper GI.
One of the patients asked that she had a patient with ulcerative colitis. Calprotectin was 660 on the GI map. A week later, the calprotectin was 150 on a standard lab. So, it's difficult to kind of compare among labs due to, we know what our lab does, and our lab certainly does everything that's required of a CLIA lab and then some. So we know that our marker internally validates very well. We can't speak to other labs in terms of how they perform the test and also do their validation, but also be aware that calprotectin is a marker that can change over time. So can it change quickly in a week?
Potentially, yeah.
Okay.
Yeah, because it's really responding to what's going on in the gut. Now, if you have chronic inflammation that's not necessarily changing, you shouldn't expect to see that changed. Generally, though, if you're doing a retest, say after a treatment, and you want to see what's happening with the levels, it's always recommended to use the same lab that you started with so you have that consistency and baseline, because otherwise you're factoring in possible methodological differences.
Yeah, I have to say sometimes this sort of question comes up when I'm managing a patient as a functional practitioner, chiropractor, and then the patient goes to see a standard GI doctor and they send him to LabCorp or Quest and get a different result, and then they kind of scoff at this test.
Right. Yeah, I mean, that's always going to be an issue when you're comparing tests. We see that, like I mentioned with H. pylori, where a patient may be negative with a stool antigen test, they do a PCR test, and because of the sensitivity level, we pick it up. And the other lab with the other method didn't pick it up. So,
Okay.
So it's ideally you're doing the same if you're kind of comparing time points with the same test.
Okay. Okay, good. Go ahead.
Okay, so on the next slide, it's just kind of a visual reminder of what we're looking at. So, we're so used to looking at tests and numbers and kind of thinking about things a bit more in the abstract when it comes to the gut because we can't see the ecosystem. It's important to really kind of come back to sort of the visual understanding that when we're talking about the bugs, if this is the lumen side towards the top, you can see the bugs here. And if these are largely representing beneficial bacteria, they're secreting factors, particularly short-chain fatty acids, that then are absorbed into the mucosa that then basically elicit effects, particularly from the immune cells and the epithelial cells. Certain epithelial cells, the goblet cells, respond by producing mucus, and then certain immune cells, the plasma cells, respond by producing secretory IgA. The secretory IgA is secreted into the mucus layer. So just kind of another quick little clinical pearl is you'll often see low secretory IgA along with low akkermansia. And that's indicating that likely this mucus layer that includes mucus and secretory IgA could be deficient. And that's you can see here, it's representing that this mucus layer is keeping the microbes away from the mucosa, which is important to keep the reactivity, the immune reactivity down. So that's part of the whole immune tolerance, immunotolerance type phenomenon is preventing overreaction of the immune system to harmless microbes. So just a little bit more about the Firmicutes Bacteroidetes phylum. Just to reiterate, they're anaerobic and they're dominant in the colon and the main source of short-chain fatty acids. And those short-chain fatty acids have many important effects, one of which hopefully we'll have a little bit of time to go through here in a little bit when I go through one of the studies, but they're definitely overall key for colon health, particularly in terms of protection from pathogens and opportunists. So it's not common to see some of these opportunists, particularly under autoimmune triggers, for example. And when we do see them, that suggests possibly the overall beneficial bacteria may be deficient. So the other information I've already talked about, so I'm going to move on. Just want to touch briefly on inflammatory dysbiosis. So this is a subset of the inflammatory, kind of the main inflammatory types of organisms on GI map. So there are particularly inflammatory bacteria in the Proteobacteria phylum. Most of them tend to be in a subclass or subgroup called gamma proteobacteria. So they're all fairly related. A lot of them have been shown to have the more inflammatory type of LPS, for example. But quickly, I want to focus on Klebsiella. So that's something we've talked about a little bit so far here. As an example, there's this really interesting study that just came out a little while ago, actually just this past summer. So the title is Intermucosal Connection Between the Mouth and the Gut in Commensal Pathobiont-Driven Colitis. So this is basically showing the connection between dysbiosis in the mouth and dysbiosis in the gut, and then what can happen in between. So they say here real quick, periodontitis leads to expansion of oral pathobionts, including Klebsiella and Enterobacter species, which are both on our test, in the oral cavity. Amassed oral pathobionts are ingested and then translocate to the gut. So they're basically swallowed, pass through the stomach, and then end up in the lower GI tract, particularly if there's not enough stomach acid, where they activate the inflammasome in colonic mononuclear phagocytes, triggering inflammation. And I want to go on to a little bit more information here. So they say in the article, this evidence suggests that at least two conditions must be met for oral pathobionts to ectopically colonize the gut. First, the colonization resistance of the gut-resident microbiota must be disrupted, often meaning that you have a lack of beneficial bacteria, enabling the oral microbes to invade the gut. Hence, intestinal inflammation favors the growth of Enterobacteriaceae. That's kind of a subgroup that includes E. coli, Salmonella, Klebsiella, et cetera, including bacteria that translate from the oral mucosa. One more thing I want to highlight from this article is alternatively, neutralization of gastric acid or inhibition of acid secretion could promote ectopic colonization of oral bacteria in the gut. For example, it has been reported that the use of proton pump inhibitors, which reduce production of gastric acid, leads to increased colonization of mouth-resident bacteria in the gut. Consistent with his observations, it's been reported that gastric acid inhibitors worsen clinical outcomes in IBD. So definitely this is an important article that kind of follows up on earlier research showing this integrative approach. You got to take the whole GI tract into account. When you see Klebsiella come up on a test, the questions you want to ask are, is there evidence it's causing inflammation? You may even want to check with the patient, ask the patient about their oral health. You may want to look at indicators of stomach acid, because it may be telling you this patient might have insufficient stomach acid. So lots of kind of connecting the dots there when you see these various organisms. So that's, I talked a little bit about the insufficiency dysbiosis, inflammatory dysbiosis. Those two are really linked. As you can imagine, when you have a lack of beneficial bacteria, that's one of the key factors that allows these inflammatory microbes to grow in the lower gut. I just want to touch on the digestive dysfunction dysbiosis. When we think of digestive dysfunctions, most clinicians go straight to this digestion section and look at the markers. Steatocrit is looking at fat malabsorption. In this case, it's high. Lastly is a marker for pancreatic enzyme deficiency or production. In this case, it's low. But if you see those normal, does that mean digestion is fine? And oftentimes the answer is no. Fortunately, we can get some additional insights by looking at the patterns of the microbes and what we know about certain microbes, such as H. pylori. So this is kind of coming back to what I talked about, that this review article basically came to the conclusion that most patients chronically infected with H. pylori have gastritis and also hypochlorhydria. Not necessarily all. It's important to note not everybody that has H. pylori has low stomach acid, but a pretty large proportion of them do seem to have low stomach acid. That low stomach acid, from various reasons, in this case, they're looking at proton pump inhibitors, leads to certain types of dysbiosis that's really reproducible. So they mentioned here in the highlighted area that Streptococcus and Enterococcus species are among the most common that you see elevated. Also, PPIs are a risk for C. diff infection. So I'll get to that. Hopefully, if we have a little bit of time later on here too. Sometimes decreased Faecalibacterium as well. So these are the patterns that we can see, for example, in relation to low stomach acid. And there's a lot of research backing this up, particularly with Streptococcus and Enterococcus. Many studies have replicated that finding that you see elevated levels of those in stool when patients have been shown to have low stomach acid. So this is just an example of a patient that had high H. pylori, so suspected low stomach acid. Again, not diagnostic for low stomach acid, but something to think about. You're looking for an overgrowth pattern. We see the overgrowth pattern typically in the normal bacteria section. This would be pretty significant. We don't always see it this prevalent with so many markers elevated, but we often do see this overgrowth pattern, particularly with the Firmicutes phylum. And then in this case, we also saw Faecalibacterium was low, which really follows that particular study that was found to be low as well with low stomach acid. Your opportunistic bacteria is really where you're going to see the most common evidence of overgrowth. That's where the Enterococcus species are and Streptococcus. So if those are high, then suspect that low stomach acid may be an issue. And then some of these others too, like Pseudomonas, can also be overgrown when stomach acid is too low, or there's other.
Can I ask about Methanobrevibacter? So we know that methane SIBO now is now EMO, according to Dr. Pimentel. And so therefore it's an overgrowth of methanogens like Methanobrevibacter, Methanobrevibacter smithii. And so if the patient has a positive SIBO breath test for methane SIBO, would we necessarily see elevated levels of Methanobrevibacter? And what if they don't correlate?
That's a good question. So they generally do seem to correlate as far as we can tell, but not always. So that is an important factor to consider, that Sometimes you can have an increase in bacterial, or in this case, microbial, because Methanobacteriaceae actually are archaea, not bacteria, kind of closely related. But and so you can have an increase in function of microbes in some cases without necessarily a significant increase in their numbers. Now, that said, on our test, and based on looking at literally thousands of tests over the last couple of years, the Methanobacteriaceae family on our test tracks very closely with overall evidence of overgrowth on our test. And you'll see here in a moment, if I get a chance to get to one of the slides here that talks about that more, it's really a direct reflection of the level of fermentation, particularly in the colon. So when you see Methanobacteriaceae, in our experience, it seems to be clinically relevant anytime it's in the E9 range, not even above the cutoff. It highly correlates with this. So generally, yes, I would say when patients have also had a breath test, they will have some level of methane detected, but it's not necessarily 100% correlation, for example.
And somebody asked a question about H. pylori, which is, can't it also increase stomach acid production depending upon where the H. pylori is?
Yep, that's true. So there is some evidence that in some cases that can be the case during more of an acute sort of early infection. And it really does depend. Apparently, there's some conflicting research on it. So it's not really all that well studied, but it does depend on where in the stomach it's infecting and the extent of the infection. Now, there is some evidence based on a couple of studies I came across that typically the infection can migrate in the stomach over time. And I forget which direction, if it's antrum to corpus or corpus to antrum, but the overall conclusion was that it tends to long-term be more consistent with hypochlorhydria. But some patients may have an increase in acid, depending on where the infection is.
Okay.
So this is just kind of showing you in this example that Klebsiella was elevated, which again is something we often see when there's other evidence suggesting low stomach acid. And that goes along with that research that suggests Klebsiella may often come from the oral cavity or possibly the respiratory tract, which is also another common location for Klebsiella. And then it may get into the GI tract through those routes when stomach acid is insufficient. I'm going to go ahead and skip this here, other than, of course, whenever you see an overgrowth of inflammatory species, we had that list higher up on one of the other slides. Oftentimes, but not always, you'll see calprotectin elevated. So there is a correlation there. We see it most commonly in, for example, patients that have already been diagnosed with inflammatory bowel disease. We often will see that connection between higher calprotectin and higher levels of inflammatory species.
By the way, if pancreatic elastase is low, is the best recommendation is to recommend that they supplement with pancreatic enzymes, or are there other recommendations?
That's a good question. So a lot of clinicians do take that route to help compensate. But of course, you want to also ask the question, what may be contributing to that?
Right.
So there may be issues with the pancreas itself. Of course, pancreatitis, things like that may play a role for some patients. But even low stomach acid can lead to lower release of digestive enzymes. That might be another factor. For a lot of patients, it can be gut-brain axis issues.
Oh, interesting. Low acid leads to low pancreatic enzymes.
It's a contributor. Yeah.
Okay.
It wouldn't be considered, if you say you have really low elastase, outright insufficiency, that's well below that 200 cutoff. It would probably be unlikely that that's the sole contributor, unless, for example, stomach acid is exceptionally low. You may be wanting to look into some other contributing factors at that point.
And if steatocrit is high, that's fat in the stool, is the best recommendation to give them lipase, fat-breaking fat digestive enzymes, or is bile going to be more beneficial, or both?
Good question. So just based on the presence of excess fat in the stool, you can't tell directly just from that which upstream process is affected. Now, if you do see that elastase is really low, particularly if there's outright insufficiency, meaning well below 200, then it's more likely that the high steatocrit would be caused by lack of lipases from the pancreas as well. But you can also have a scenario where there's insufficient bile. What's probably less appreciated is issues in the small intestine, which is where fats are primarily absorbed, where they're supposed to be absorbed in a healthy gut. So if you have an inflamed small intestine and you're not able to really absorb fats as efficiently.
Such as if you have SIBO?
Exactly.
Okay.
Right. Just have a few more things to go through, and then based on time, do you want me to stop here or take more questions, or do you want me to continue on for a couple more slides?
Go a couple more slides.
Okay. So again, these are the three main dysbiosis patterns. That learning to recognize them can give you a lot of insights into what's going on in the overall gut in terms of physiology and the microbiome, and even some insights into, say, lower versus upper GI issues. Kind of skip this, but we all know that, of course, pH is one of the key factors along the GI tract. We mostly think of stomach being the key kind of checkpoint. So when you may unfortunately ingest pathogens, for example, food poisoning, if you have good stomach acid, then they're less likely to survive the transit. But what's less appreciated is that slightly acidic levels in a healthy colon, especially the ascending colon, are almost as important in helping to keep pathogens and other opportunists from colonizing the colon. So just going to go through a quick example of that here. This is a really interesting paper that was published recently. So the title is Inhibiting Antibiotic-Resistant Enterobacteriaceae. So that's in the Proteobacteria phylum. That's a particularly inflammatory group. So inhibiting that group by microbiota-mediated intracellular acidification. So just to kind of cut to the chase, they mentioned Klebsiella, E. coli as kind of the examples they're looking at here. And then the highlighted in orange part towards the bottom says Klebsiella pneumoniae, Escherichia coli, and Proteus by acidifying the proximal colon and triggering short-chain fatty acid mediating intracellular acidification. So essentially what they showed in this study is that normal levels of beneficial microbes that are producing adequate levels of short-chain fatty acids literally inhibit the growth of these pathogens by basically producing the short-chain fatty acids that then are taken up by those cells, and that acidifies the cells and basically prevents them from growing. So that's one of the ways in which these beneficial bacteria like Clostridia, Bacteroidetes, can help to keep those pathogens in check. So once again, when you see those at high level, particularly Klebsiella or Proteus, that's one of the things to be looking at is evidence for low beneficial bacteria. They actually showed in this study that just restoring the beneficial bacteria and the short-chain fatty acids can be enough to address those pathogens. So that's kind of an example where you don't always have to resort to antimicrobials. Restoring gut balance may be sufficient.
Can I just ask a question about antimicrobials? I interviewed Dr. Jason Haralick a few months ago, and he mentioned that he thinks that taking antimicrobials like berberine can significantly damage the microbiome. Do you think that that is something that is liable to happen? Do we see evidence of that? In other words, can natural antimicrobials produce damage in the microbiome? The way antibiotics can, say, for example.
Generally, no, we have not seen that, not even close.
Okay, good.
Mostly when we see a really broad deficiency pattern, it's related to antibiotics or other pathology like IBD or something like that.
Good.
Real quick, though, I think he was referring to, at least in terms of research I'm aware of, berberine in one study a few years ago showed that it can, long-term or high doses, can reduce diversity but it was in an animal model in mice. So that suggests, maybe not taking super high doses for a long time would be.
By the way, what's a high dosage? Because, for example, I'm using berberine not only for gut patients, but we use it to help manage metabolic factors, and I also use it as a sort of a substitute for metformin in anti-aging programs.
Right. Good question.
It works synergistically with metformin to manage blood sugar. So,
right. So that was really just one study, and I have not come across any additional studies on that. Again, it was a mouse study.
Okay.
I recall that it was a very high dose, though, something maybe equivalent to at least more than 5 grams per day for humans. Okay. So, again, you have to take those kind of things into account, that if it was just an animal study and it was super high dose, maybe doesn't apply to real world settings.
Right. Okay.
But that has not been our experience. The patients on strong antimicrobial botanicals don't seem to develop a deficiency pattern based on what we see on GI-MAP so far.
Right, good. And that's been my experience as well.
Okay. So this actually, it's kind of interesting that we talked about that, because I won't go through this full case. It's not really a full clinical case, but just a quick example of an older female, history of bladder cancer, was on long-term amoxicillin antibiotic for 5 or more years. Main complaint was just chronic diarrhea. So of course there was a suspicion of antibiotic-associated diarrhea in this case. You can see here very broad deficiency, which would be pretty much expected from such a long-term exposure to an antibiotic. But just based on what we talked about before with the insufficiency, you would expect that this would potentially lead to overgrowth of opportunists or pathogens. And sure enough, you see Pseudomonas and Klebsiella overgrown. And it turns out that those two are among the ones that are known to be resistant typically to amoxicillin. So, we're seeing exactly the pattern you'd expect based on that patient being on that antibiotic. And this kind of scenario to me is very important. You're thinking about, which gut test you want to go with, and how good is a gut test. And you can kind of debate, validation approaches and all these things, and sequencing versus PCR. But in the end of the day, it really comes down to, does this clinically validate? And if you know a patient, for example, is on an antibiotic long-term, you would expect to see patterns like this. If you don't see patterns like this, there may be some individual variation. But so that's just something to keep in mind as you're going through, some people may try different gut tests. You want to make sure that the test you're using clinically validates and that it makes sense based on the patterns that you're seeing.
Can I just comment? Your test looks at if there's pathogens, what antibiotics might be effective for those pathogens. Some of the stool tests, at least in the past, I know, used to use natural antimicrobials. How come you don't include that?
That's a good question. So I'm not sure of all the reasons why. I know part of it is we wanted to kind of keep our test to molecular. That's what our lab does. That other sort of approach to look at potential sensitivity involves culturing. So that's part of the picture, but also, how realistic is it from a couple different standpoints that what you're measuring on a Petri plate with one organism really translates to how effective that's going to be in an actual vast, complicated ecosystem. You don't really know that. Plus, most clinicians tend to use common formulations. They're not just, some do use single ingredient approaches. A lot of clinicians use, formulations that they found to be generally effective. So it's not necessarily going to affect what they use clinically either. So it's not always clinically useful information.
Okay.
So I think I'll skip all of this here. Just real quick, and one more thing about Klebsiella and also Morganella. They have been linked to excess histamine production. So again, be thinking of these things when you see, Klebsiella overgrown. You want to kind of look at the context. Are beneficial bacteria lacking? Does the patient also potentially have histamine intolerance symptoms, et cetera? So the more you know about these microbes, the more you can potentially clinically connect those dots. this is the only thing I was going to show, and I'll stop with this one. This is a little bit of a complicated diagram, but it's representing the colon. On the left side is the proximal colon, and then moving on across horizontally to the right towards the distal colon. There's a lot of research showing basically the dynamics in the colon so that you understand better why a patient may have, say, a high Firmicutes, low Bacteroidetes, why they might have sulfur-reducing bacteria or methanogens. So the main point I wanted to make here is generally in the first part of the colon is where most of the fiber fermentation, carbohydrate fermentation is going to happen. That can release hydrogen. And then basically that part has to kind of start happening first before that hydrogen is available for the methanogens. And then basically further down the line would be the sulfur-reducing bacteria, which tend to be more in that latter part of the colon, where also Bacteroidetes tend to dominate. And that's going to be affected by transit time and other factors too. But there's a lot of sort of modeling that's being done to understand the ecosystem. And I think this is a great visual to understand why do we see high methanogens on our test when there's high Firmicutes that are showing you that there's a general overgrowth patterns, probably because they're fermenting, generating H2, and then promoting methanogen growth. All right, so that's a few additional slides. A lot of material that we could always cover. Maybe we'll have an opportunity to do a part 2 at some point.
Okay.
Yeah, so that brings me in. So really it's all about connecting the dots and just learning. We have a lot of educational information. Of course, we offer the consultations, so I would encourage everyone to take advantage of those resources because the more you can connect the dots, the more you can get out of the stool test in terms of good clinical insights that can really help you be more effective in helping your patients and clients.
I have one more question I'd like to ask, and then I'll see if anybody else has some additional questions. I've interviewed several clinicians, one of whom is very prominent and feels that some protozoa like Blastocystis hominis and D. fragilis are commensal and not something that we should be concerned with. And you know, he's been talking about that quite a bit. And I think it's common for a number of functional medicine practitioners to see that on a GI map stool test and say, here is likely the cause of your problem, and are used to treating it and getting results. And then I also talked to, in another recent podcast, another practitioner, Alana Gorovich, and she felt that in patients where it correlated with their symptoms, very important to treat blasto and D. fragilis. What do you think? Are these pathogens, are these commensals, or does it depend?
Good question. So they're not likely to be commensals based on our experience because we don't see them in the majority of patients. Commensals tend to be present in the majority of individuals. I think the real answer is probably somewhere in between. So if you look at the research, most of the research suggests that Blastocystis in particular, and possibly Dientamoeba, their conclusion in most of the research is that for the majority of people that have them, they're not a problem because most of those people are, quote, asymptomatic. But I think they're thinking of them asymptomatic in a conventional sense, not a functional sense. So they may be ignoring SIBO and these other things, mild GI symptoms, et cetera, and not taking those into account. With GI map, we only see, I would say, probably more than 95% of the time when we see Blastocystis and or Dientamoeba fragilis, it's along with that digestive dysfunction pattern. I don't think I've ever seen Blastocystis just sort of by itself with no other patterns where it's kind of like the obvious cause of things. It's almost always in the context of things like H. pylori, maybe Candida, general overgrowth. So the question is, what's causing the symptoms? Trying to pin it on an individual organism, some may be more important than others. H. pylori often is because of its effects on stomach acid. Candida often is, because we know it can cause inflammation, leaky gut, Pseudomonas. Blastocystis, I think, is somewhere in between. Probably for some patients, if they have a certain subtype, and it's at a really high level, that may be more significant than if it's there at a low level.
Okay. So one question that came in is, what causes insufficiency dysbiosis? Is it low stomach acid?
That's a good question. So recent research indicates that antibiotics are often a major cause, even long-term, because what can happen is antibiotics can actually affect the mitochondria in the cells that line the colon in a way that makes the gut environment less favorable to those beneficial bacteria. So that's at least one example of pharmaceuticals that may have that effect. Certainly lack of fiber in the diet. That's been correlated with lower levels of these beneficial species. Any source of inflammation. So inflammation is highly detrimental to many of those beneficial bacteria. So whatever gets inflammation going, that can really turn the tide. One of the other contributing factors, and it's related to low digestion, is too much protein in the colon. That can be from just high protein diet or not digesting well. But this process of sort of breaking down amino acids, which is called protein fermentation, and then breaking down carbs, which is carb fermentation, so primarily fibers, they're antagonistic. So the less fiber you have and the more protein you have in the colon, that can really also be detrimental to those beneficial bacteria.
So another question is, how does the ketogenic diet, and I would even throw in there, how about the carnivore diet, impact the microbiome?
I've actually only seen 2 cases so far, and they were almost identical. And they were probably they were both among the worst cases I've seen as far as dysbiosis. That's a perfect example of this sort of, these sayings of test, don't guess, or the opposite of that is treat the patient, not the test, right? 2 different philosophies or ends of the spectrum. When you see that, so in both cases, the patient improved in symptoms. They had kind of typical GI symptoms, gas, bloating, apparently issues with carbohydrates, so they went full carnivore. Symptoms improved, and yet you look at the gut microbiome and it looks terrible. Lots of inflammation in both cases, just massive dysbiosis. So the question is, well, is short-term improvement in symptoms, are you setting the stage for long-term problems? And is there another way to address those symptoms without kind of ruining the microbiome? So good question. I mean, I guess time will tell. From everything we know about the microbiome, those results look terrible. They really would be considered by almost everybody as really bad dysbiosis.
Okay. And one more question. Is it necessary to stop digestive enzymes before collecting the stool sample? In fact, are there any other recommendations that should be taken before collecting the stool sample?
Not really. A lot of it's going to depend on the clinician goals in terms of what you want to see. Do you want to see how your patient is doing on the treatments, or do you want to see them, of course, before treatment, et cetera?
Well, I guess if they're taking enzymes, would that affect the pancreatic elastase levels?
There's not much evidence that does, no.
Okay.
Certainly with elastase, that's typically not in the enzyme formulation. So, you wouldn't literally affect it from just coming from the antibiotics or the main one. If you do take antibiotics, we recommend waiting at least 30 days post-antibiotic treatments, maybe 60 days, to allow the microbiome to recover somewhat.
Okay, excellent. Thank you so much, Thomas.
It was my pleasure. And thank you.
Great. And thanks, everybody, and we'll see you next month.